In this lesson, we're going to see how T cells work with B cells. And in a lot of textbooks, this is judged to be one of the most important parts of immunology. It helps us understand some diseases like HIV and autoimmunity. So this is given a lot of emphasis in textbooks. Now, to prepare for this drawing, I watched hours and hours of medical lectures online. I read website articles from scholarly journals. I watched educational YouTube videos trying to get the most up-to-date information about this to be as accurate as possible. Now, some time may have elapsed between me filming it today when I'm filming and when you actually watch this video. Maybe several years will have gone by. And there's a possibility that some new information will have come to light in the interim, and they may be backing off on this and going, well, there's parts of it, it's generally right, but we know a little more about this and it's even more complicated. This is a general guideline of the way it works. And hopefully it will let you look at things like this. I found this chart on Wikipedia. This is one of the easier resources to use. I was very happy to find this because these pathways are exactly what I was wanting to find. So this is considered to be a very simple and easy chart. So I have taken this information. This is almost exactly what we're going to do in this lesson. We've got cute little cartoon characters, a little storyline that will hopefully be easy to follow. Now, just because we're using cute little cartoon characters doesn't mean this is kid stuff. I actually got this idea of using cartoon characters from a medical doctor who was doing lectures for medical students, and he was using cartoon characters. Okay, so we're going to start out with this macrophage right here. By now you probably know this shape to be a macrophage, hopefully. And we're going to review what the macrophage does. Get a little closer, there we go. Okay, so let's read this right here. The macrophage, or dendritic cell, we're just using the macrophage here, presents a piece of pathogen to a naive T helper cell. So let's draw a pathogen. I'm going to make my pathogen orange. I don't know why I take it to making pathogens orange, but all right. So this is a non-specific pathogen. Could be a bacteria, could be a virus, could be a protozoa, could be a fungal part, who knows. And the macrophage engulfs it and is then going to present little pieces of it on the MHC2 clip. Now let's go ahead and give the macrophage a big mouth to help us remember he's the big eater, and some cartoon eyes that are kind of looking over here at the T cell. And the other internal part that we're very concerned with are the lysosomes. It's got lots and lots of lysosomes because it is a professional eater. And it's going to use those lysosomes to digest this little pathogen into little bits and then present a piece of it over here on an MHC2 clip. So this is the MHC2, and it's really called the major histocompatibility complex, but I like to think of it as my house cleaning. And the number 2 version is found only on these antigen-presenting cells, macrophages, dendritic cells, and we'll see in this lesson sometimes on B cells as well. And as long as we're at it, let's give the macrophage some other things we know, like the CD31 hook. This is the thing it grabs neutrophils with. Now, of course, massively out of scale. It'd be teeny tiny, too small to see, but we need to see it. And there'd be some other receptors. Any shape at all will do. And of course, there would be some MHC1s too, because it's a body cell. It would have a few of those. It would have other MHC2s. And if you want to label it, can. You don't have to. Okay, so here it is presenting a piece of that pathogen on its clip. And the entire point of doing this is so that this T cell could come over and look at it. Well, not really look at it, but feel it. So this is a naive T cell. Naive just means it doesn't have any experience yet. It doesn't know what to do. It doesn't mean it's stupid. It just means it's not quite sure what to do. And let's put helper on its jersey. And then CD4, because remember, the CD4s are helpers and the CD8s are the killers. And give him kind of an innocent face, like that. Doesn't quite know what to do. Not sure. And it comes over and checks this thing out with its TCR, its T cell receptor, and I'm drawing them as kind of a Y shape just to remind us that it's similar to the ends of antibodies. Just a little reminder picture here. Zoom out just a little bit here so we can see both of them. Okay, so this is like a Y of an antibody, and these would be the places where the antigens stick, kind of right in here. Now here's the T cell receptor. Look at the similarity here between that and that. Right, very similar. Now, it doesn't actually have a Y shape, so sometimes they draw this receptor like this. So you can do that too if you want. That would be fine. Or sometimes they go like this. That's kind of very similar to an antibody. So if you want to do that, that's fine. Back in here. I just think it looks more like a little hand. It kind of draws a kind of very slight Y. So whatever shape it is, we just need to know that its job is to feel these antigens. And of course, there are millions of different CD4s floating around. They all have a unique shape. Each one has just one shape here. So most of the T cells that come and bump into this won't match. They'll come, they'll feel it and say, doesn't mean anything to me, and leave. But hopefully, the correct match, this guy actually has the matching shape for this little piece of pathogen. And so it'll stick, and he'll go, whoa, this is something I recognize. This is something that is a bad thing, I think. So now he's locked on to this, but he needs to know what to do, because he doesn't know where this pathogen came from. He doesn't know whether it was hiding inside a cell or whether it was just floating around in the blood or in the interstitial space around the cells. And that makes a big, big difference where you're going to go to look, because the job of this T0 guy is going to be to either call the T killers to come and help or to go over to the B cells and get them to help. And if he chooses the wrong one, then it'll be ineffective and the army will be looking in the wrong place and the pathogen won't get killed. So this is very, very important. So how is this guy going to know where to look? Well, the macrophage has to tell him. And of course the way cells communicate is through chemicals, cytokines. So he sends over cytokine messages. And they don't really understand exactly how this works. There's still a lot we don't know about this. But we do know that the macrophage is probably responsible for telling this guy what to do. So the message will be either to go look inside the cell or look outside. So looking inside, let's make it over here, look inside the cells. And let's make this option look outside. Now at this point I want to give you two very important terms. For us they're just going to be little things in parentheses here, because the main point that I want to make is you either look inside or outside. That's the most important thing, is to understand that. However, you're going to see this term in any book you read on this. They're going to say cell mediated response. And when you read this in a book, it's going to sound hard and complicated. They could have just said look inside the cells and you'd understand it, but no, they have to use the term cell mediated response. And that just means kind of like hand-to-hand combat between cells. You're going to go get the killer T's to actually attack, cell-to-cell attack. And this pathway is going to be called the humoral response, based on the word humor, and we don't mean that it's funny. What I want you to do is put a little asterisk. I'm going to use, like, I don't know, maybe blue. You know, an asterisk is one of these little like star thingies like that, right after the word humoral. And then down here, this is the note that explains it. Right there. It's the only place I could work it in on the page. Humor is a word from the Middle Ages. It means body fluid. Therefore, a humoral response occurs in the body fluids. In other words, antibodies get dumped into a body fluid, usually into the blood. And I have two little pictures from the Middle Ages to show you. There's one showing the 4 humors. So this word is a very old word from back when we knew very little about the body. And they thought there were 4 basic body fluids. There would be phlegm and blood and yellow bile and black bile, I think. And the amount of each one of these body fluids you had, so they thought, determined what kind of illness you had, and even determined personality. So here's a funny little chart. It says choleric, and so they would have a certain attitude in life. The sanguine people, they have like a lot of blood, and they would have a certain personality. The phlegmatic and the melancholy. So their explanation for these personality traits was just the amount of bile or blood or phlegm you had. Now, of course, that's laughable and ridiculous to us because we understand that bile made by your liver, its job is to help digest foods. It has nothing to do with your personality. But, you know, they didn't have electron microscopes, and they didn't have hundreds of years of research to look at. So this was a wild guess. Sometimes science gets it wrong. And here's one place where mainstream science got it wrong. They were on the wrong track. So that's where the word humor comes from, the historical origins. So when we say humor, we mean body fluids, in this case probably blood. So the humoral response will occur in the body fluids. But we're going to do this cell-mediated response first. So let's look over here at this guy. When T0 wants to look inside of cells, it changes slightly. It differentiates, makes copies of itself, and all these copies will be number 1. See, so this is helper. Still a helper, it's still CD4, but now instead of 0, the naive, it's number 1. It's got a big smile on its face now because it has a job. It's not wondering what to do anymore. Give it some little receptors there. So it knows what to do about that little thing that it just felt up at the macrophage. So it's going to do several things. One of them is that it's going to send a message back to the macrophage again. So there's no significance to the fact that it's coming out of the head. I mean, this is just a cell. I could have drawn it coming out of any place. So it's going to send cytokines back over to the macrophage, telling it to speed up. It's going to be like, go macrophage, go, go, go. eat, 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 process, 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 get those antigens out. And just for you guys that like trivia, if you want to know what kind of cytokine it is, this is interferon. And these cytokines, called the interferons, got their name from when they were discovered. It was in the process of interfering with the viral process, so they got named interferons, and then they discovered, wow, interferons do other things too. That's very often the case in biology. Something is originally discovered in one context, and that's where it got its name, and then they discover that it does all kinds of other things, and the name doesn't make so much sense after that, but we're stuck with it. So these interferons go over and tell the macrophage to speed up, and it also, strangely enough, sends a message to itself. So let's put some dots down here going like this. Now there's a special complicated name for this, which we're not going to bother with when they send messages to themselves. So right here, TH1 also sends messages to itself. That's what's going on here, telling it to start making clones. So it says, self, we're going to need an army. We need to go through mitosis and make a whole bunch of ourselves. So its replica, one of the replicas will be down here. So this will also be number 1. Let's give it a mouth that's like saying something. Sort of, a little bit. And it'll be number 1. It could also be looking, I'm going to make my eyes looking over here. It'll make more sense if its eyes are kind of looking at its neighbor here. And it's still a helper. And it's going to be talking to this guy. This will be the killer. So I'm going to write killer on its jersey and CD8. Often they're just called CD8 cells, but its abbreviation is usually T with a little c, C standing for cytotoxic. Because remember I said before that though they've been called killer T's a lot, nowadays they're kind of preferring to call them cytotoxic T's. So he is wondering a little bit here what to do, because he has found a little antigen that makes sense, fits its receptor, it's locked onto this, and it is being presented on, now can you remember here, what does the killer cell match up with? The helpers, the CD4 matches up with those antigen presenting cells, the macrophages and the dendritics. The CD8s have this little extra protein that reaches over and recognizes MHC1, and where do you find that? On body cells. So this would be a body cell. So let's assume this is an infected body cell. And what's being presented is kind of one of these bad particles that's floating around. So this killer cell is thinking, or maybe he's saying out loud, you could put a little quote bubble too, it doesn't have to be a cloud. He's thinking, should I kill this cell? So interestingly enough, the killer doesn't immediately kill when it latches on. you'd think that that would be the way it would work. You latch on, whoop, I found something, I know this is a bad particle, it's on a body cell, I know that shouldn't be there, bang, kill it. However, it's a little more complicated than that. It doesn't immediately kill the cell. It waits for confirmation from a helper T. Because after all, killing body cells is a serious thing. You know, we don't want to mistakenly be going around killing good body cells. So it needs confirmation from this helper here. So it's asking, should I kill this cell? And the helper number 1 is going to say, go for it, kill. Right? And how does it communicate? Cytokines. So these are cytokines. And for you trivia buffs, if you want to know, this is interleukin 2. IL-2, in this case, now it has some other jobs also, but when it's secreted here, IL-2 says, go, kill. You know, I might add a little yellow here to kind of make that show up. In fact, if you want to add a little yellow to your other ones, I might stop and just kind of add some yellow to all my cytokines there. Make them show up a little bit. Oops. Now, one interesting side note about this stage right here is that this is one of the things that goes wrong in HIV/AIDS. The AIDS virus attacks CD4s. That's the only type of body cell that the virus will attack. And so these guys get infected or killed. So if we remove him from this process, what would we have? Well, we'd have an infected body cell. Let's say you come down with the common cold, right? And here comes a killer cell over, matches up, and it's thinking, should I kill this cell? Should I kill this cell? And there's no CD4 there to tell it to go ahead and kill. So though it locks on, it never kills. And so the virus goes right on replicating, and the body cell dies. So killer cells are very, very dependent on these helpers. So now you understand a little bit how HIV works. So let's see how they kill. Actually, what we need to do here, we forgot to make him a perforing gun. Put a little gun right there. So he's not going to get the gun out and shoot unless he's told to. So once that cytokine message goes across, IL-2 says get out your gun and kill the cell. What happens is that he turns into an active killer and put his little gun in his hand. Now, of course, it's not really a gun. What it is little granules filled with perforin, and they just release the granule, just like a mast cell releases its granules. It would just kind of be the same. It would just release these. And so he's got a smile on his face, but he's kind of pretty serious. You can make his eyes looking over here too. He's in killing mode. TC killer CD8. And it looks like his gun is going off here. If you make a couple little things there, kapow. And let's make a note here. Shoots perforins and something called granzymes. And we'll do more of that right in the space here. So what does the hole look like? Well, it's kind of just, you kind of make a little oval here and make the oval consist of little circles like that, and I'll explain that in just a second. So the little perforins are going to go and make this little hole. It's like insta-hole. They just kind of assemble to make the hole. And then also, there's two kind of bullets here. There's these little granzymes that then go into the hole. Okay, so there's also little dots that go in. Now, before we go on and explain the perforant, let's just go back here and pick this up and let you know that a few of the clones will go to sleep, holding on to their little antigen maybe, okay? TC mem. And a few of them will become memory cells. Most will become active killers, but a few are going to be saved so that in case we meet this pathogen again, they're ready to go. However, and of course this would be just mitosis, so let's write mitosis makes lots of killers. T cells don't multiply unless they're needed, and then they make lots and lots of clones. Okay, let's talk a little bit more about this, because you're probably very interested in how this perforin gun works. So let's draw the edge of a cell right about here. You can't see the whole cell, just a little piece of it. And then right here, draw an oval. And make the oval, let's see, let's erase the middle line there. Make the oval have little ovals, like this. And then draw two lines down like this. Another kind of curvy line, same curve as this one. And then little lines going down. And then at the bottom of each of these, kind of make it round. So they're going to look like cylinders. And then if you want to be fancy, you can kind of shade a little in the middle, but you don't have to. Now let me show you a picture of what this really looks like. Here is a computer-generated image. Now these would be the little cylinders we drew. They're more complicated than a cylinder. We've kind of simplified them. They're kind of a funny shape, and you can see up here they've tried to show you a little better what that shape is. It's like they've taken this one and cut it in half. So you can see on the inside, each piece, we just kind of drew it like this as a cylinder. It actually has these kind of bottom pieces sticking out. And here is a kind of a protein map of that little piece. You can see the alpha helices and the beta sheets. And there would be that thing sticking out. Very clearly here, though, you can see that it's got a central hole. Now this is the best we can do at a photograph. This is an electron microscope picture showing down to the molecular level. Okay, so these would be like molecules. Maybe they're the tops of the heads of the phospholipids. But it does show clearly that they are circular. Here's some more pictures again. That's the clearest we could possibly get in terms of a photograph. And it's not even a photograph, it's an electron microscope picture, so it doesn't use light. It uses a different method of recording the image, so we can't really even call it a photograph because it doesn't use photo. And lastly, here is a picture again showing a really up close view. These are all the heads of the little phospholipids and the tails in the middle here. So that gives you a scale of how big this thing is. And each of these would be a little perforin unit, one little unit, the little cylinders we drew. And the neat thing is that nothing puts them together. When they fall down and hit the membrane, they just automatically kind of do this. They self-assemble. You don't need a little enzyme robot to put them together. So let's make some individual units kind of coming down. So the T cell that shot these out would be, up here. We could pretend it's this guy here. And let's draw some little dots here for our granzymes, because several different things come out of these little vesicles. And one of them is granzymes. So these would be granzymes, kind of like the word enzymes, but with gran on the front of it. Draw a few more of these. A few little granzymes floating around. Might even color mine green or something. Green, with green zymes, green granzymes. And let's note here that granzymes are, I'm going to put toxins in quotes, toxins, because usually when we think of a toxin, we think of like bee and wasp spray, it's toxic and it kills bugs, or something like a poison dart frog poison or something that is really a neurotoxin, and it affects your nervous system, and that's how it kills you. This doesn't have that mechanism at all, and we'll talk about the mechanism in just a minute. The granzymes are toxins that make the cell kill itself, commit suicide basically. And if you want to know the correct word for this, you can put it in parentheses. It's called apoptosis, apoptosis. Some people say apoptosis, but a lot of people say apoptosis. And let's put a note over here. Point to perforin. Perforin units self-assemble. That means they don't need any help. They just go together automatically into ring. And we could put a note here too. This is the cell membrane. And if you want to, you could even add all little phospholipid heads. You don't have to, but it gives you an idea of scale. So this is really the basics. All you need to know is the little perforin proteins assemble themselves into a ring, so they open up a hole, and this might actually help to destroy the cell. Some stuff might leak out, but mainly the job of the hole is to allow these little granzymes to go in. So this is a diagram of exactly what's going on. This is the T cell, and of course it doesn't look like a letter T. It's round, it's a cell. And this is the little vesicle that contains the perforin and the granzymes and a couple other things that we're going to just ignore. This is what I'm calling the gun. It's actually a little vesicle. It comes to the edge of the cell. It does exocytosis, so it spills its contents outside the cell, and all these little parts float around till they hit the cell membrane, and then the little perforin units self-assemble into a little ring, and then these little granzymes go through. Now once the granzymes come into the cell, then we have sort of a little cascade of things happening. Something affects something, which affects something else. So granzyme B goes over here, and it causes this molecule to come apart from this one. You see how these are together? Well, it can separate these, so this orange one comes over here, and it sticks to this thing, which is on the edge of this organelle, which I hope you recognize as a mitochondria. And that allows these little molecules to escape out, and this can also go, it looks like, and stick to this one and let some more molecules escape. And then these molecules go over and they stick to this thing, which is called APAF1. And so these little cytochrome C molecules stick here, and they create this molecule, which then can go over here, and it can activate this molecule. This is called pro-caspase 9. You don't have to remember any of these names. I'm just trying to impress you with how complicated this is. And so this molecule turns into this molecule, and then this caspase 9 goes over here, and it can affect this molecule called pro-caspase 3 and turn it into caspase 3. And caspase 3 is actually important enough to have its own Wikipedia article, and you can go read more about it there. And what it does is it comes over and it can divide these two molecules. Well, this is kind of a big clump molecule, but the red side here is acting as a safety clip. If this yellow thing were allowed to just go free, what it would do would be shred DNA. And so this is acting like a clip. Well, caspase 3 takes the safety clip off, and so here, see down here, see we have the yellow molecule. It's actually down here in the nucleus, and here's the DNA. And so this thing is like a shredding machine, and it immediately goes and starts shredding the DNA. And once the DNA is all chopped up and unusable, the cell won't be able to make any proteins, and basically it'll die. Now, I know there is a cell that doesn't contain DNA, and that's the red blood cell. That's kind of a special case. In general, once your DNA is gone, you're dead. However, it's kind of a slow death, a little bit. I mean, you know, if you just had this hole blown in this and just burst everything out and just kill the cell in a very violent way and have everything fly everywhere, it would kind of make a mess. And that might not be so good. That might disrupt things and spread little particles around. You know, there might be nasty things hiding in here. Remember, the T cell determined that this is an infected cell, and there might be some things in here you want to contain. So this slow pathway called apoptosis allows for kind of controlled cell death. Now, of course, it happens here in this T cell reaction, but apoptosis in general isn't always a bad thing because of a pathogen. Remember, in embryology especially, you have areas like between the fingers where you want the cells to disappear and open up a space. So this form of cell death can actually be a part of life. So that was one option for this guy, to turn into T1 and go call the killers so they could come get their perforin guns. The other option is to go talk to B cells. And we'll get to these other options in a minute. There are a few more options, but these are the two major ones. So the other option was to go look outside the cells, the humoral response. So this one is going to be number 2. And it's a helper, CD4, and now has a big smile because it knows what to do. It's not naive anymore. And what it is going to do, give this a little bit, is go talk to the B cells. So let's draw the B cell first. It's not quite sure what to do because it found this thing. Let's give him a little, looks like an antibody sticking there. Okay, so they can express antibodies that then they can take in. They can pull this inside, take it over, and stick those little pieces onto an MHC2 clip. Remember we said that the antigen-presenting cells, the APCs, are macrophages, dendritic cells, and sometimes B cells. So this is actually MHC2, just like the macrophage uses. And here's this little piece. So it's got this little thing, and it is saying, this little conversation bubble, saying, I found this thing. Should I make IgGs against it? Should I make IgGs against it? Now this is an important question because this B cell is making IgM antibodies right now against this pathogen. It knows this is something nasty and it needs to start making antibodies against it. What it's been making are IgMs. So right down here, make 5 little sticks joined together like this, and then the little Y at each end. So it's like 5 antibodies joined at their base. And we can even put some little antigens on a few of them to show that they're antibodies. So this is called IgM. And I think this is easy to remember because the M, the letter M, is kind of pointy and it kind of reminds me a little bit of this shape. So I find that's the easiest one to remember. An IgG is just simply a Y. That's the antibody we usually see in pictures like that. We usually see IgG typically. So it would like to know whether to stop making these and start making these. Now once it makes that switch, and this is called class switching, once it makes that switch, it can't ever go back and make IgMs. Now why did it even make IgMs? What's the good of this? Well, at first, this shape is good. It's a little bit large and clunky, but it's kind of good at picking up things. Just the geometry of this is good for when you start out. It just is more likely to pick up the antigens. But in the long run, this shape isn't so good for making a lot of. This shape is better for the long-term fight. So if you have an infection and they check your blood, they're going to test for IgMs and IgGs. If you have a whole lot of IgMs, the doctor is going to say, well, it looks like you have a very active infection. This is recent, it's current, it's ongoing. If they find a whole lot of IgGs, this tells them there's already been a switch. So this infection has been going on for a little while. It doesn't tell you how long, but a whole lot of IgGs suggests basically that this fight has been going on for a while. And in fact, you can have IgGs floating in your blood for weeks or months or maybe even years after the actual infection. So this is one diagnostic tool that doctors use to try to figure out what's going on, how much IgM you have compared to how much IgG. So the B cell is asking, I found this thing, now should I switch and start making these, which are actually a little more effective? And he's not going to do anything until he talks to a T helper about it. So here's our little helper number 2. And his mouth is going to be saying, yes, switch and start making IgGs. So actually, he's probably reaching over here. He may be feeling this and going, oh yeah, I recognize that. Yep, that's something that I know. I think you need to start making IgGs to it. So how does it communicate? Cytokines. Cytokines say yes. Just gonna make mine just a little yellow. There we go. So once this message is given that yes, B cell, go ahead, start making IgGs, then it is going to produce a lot of clones, and most of the clones are going to be this kind. This is lots. Most. We're going to go this direction. And this type of B cell, we have a special name for it. We've learned this before, so this is review. It's called a plasma cell. I want to write short-lived plasma cell. And we even looked at a picture of one. There. And you can tell a plasma cell because it starts getting all these little dots in the cytoplasm. And it is making antibodies. Lots and lots and lots. Cranking out thousands, probably millions of them. Now they come off, so we're going to make a lot of these kind of streaming away from this B cell. And I found that it's easiest, I think, if you make little V's first and then you put the sticks on. I don't know, I just go faster when I do that. But you can make them any way you want to. And these are all IgGs. If they were IgMs, we'd have to draw this little thing. We'd have to draw a whole bunch of these, so I'm glad we don't have to do that. The IgGs are much easier to draw. The other option, so this is just a few. Go to sleep. So there he is with his and he's a memory. So this is very long-lived memory cells. And this is what gives your body its ability to be what we call immune to a virus or a bacteria. So once you've had, let's say, chickenpox, then you would have a lot of these floating around. Once your body had fought it off, we did this also, but then a few of these chickenpox B cells, memory cells, would hang around for a very long time, hopefully the rest of your life. And anytime in the future we have this going on, immediately we have, hey, wake him up. Wake him up. He knows how to do this. He knows how to make the right one. So this will happen so fast that you'll never actually, quote, come down with the pathogen. It'll be fought off so quickly that you'll never even get sick, and you won't even know that the fight ever happened. So an interesting side note about this would be that we can artificially produce these. We don't even need to have a real pathogen. When you get a inoculation, a vaccine, they give a little bit of this to the B cells, and we have this taking place through the vaccination. We have memory cells formed, and you never do have to fight off the illness. You never do come down with chickenpox. You just get the little chickenpox things, and it makes memory cells, and the memory cells hopefully give you lifetime immunity to that pathogen. Now let's go back up here and finish the drawing with a few other options, things that could happen. One thing that this cell could also turn into is something called a regulatory cell. Now these used to be called suppressors, but now they prefer regulatory, so we're going to call him reg. Regulatory. Alright, so he's going to have a happy face because he's got a job now. And I want you to make him holding an object here. Just put a little hand out, put a long stick. He's going to be holding a stop sign. So here's your challenge, is to draw an 8-sided stop sign here. Doesn't have to be perfect. You can write the word stop in it so you know what it is. If you want to, you can trace around the outside with red. Okay, so the regulatory helper will tell the killer T's when to stop, because cells don't have brains, they don't know about timing, so eventually those killer cells are going to have to stop killing. The regulatory cells tell the killers to stop. So these help get your immune system calmed back down after it's had a fight with a pathogen. So again, they used to be called suppressors because they thought they suppressed the immune system, but now they prefer to call them regulatory because they just help regulate it. So another option could be number 17. So this is helper 17. He's happy because he has a job, and he is going to be holding an arrow, because that's going to help remind us that this one increases inflammation. M is inflammation. So this guy used to be seen as bad. They thought that this was something going wrong with the immune system, but now they're pretty sure that this is actually necessary sometimes to have these. Now if you get too many of these and they go around increasing inflammation all the time, that can be a big problem. But they do have a role in the normal functioning of the immune system. So how about number 3? What's the last number we're going to do? We're never going to know what 4, 5, and 6 are. I'm not even sure that they exist. He's happy because he has a job. And number 3 isn't going to be holding anything. Well, I guess we could make a stick with a Y on either end. This is an IgA. Okay, so and we can draw it down here. An IgA antibody, make a little circle there. It's like 2 Y's stuck together at the base. We could put an antigen here so we know that's the antibody. Right, 2 of them stuck together like this. So are they going to be able to stick to body cells and cause trouble with mast cells? Nope. So since these don't stick to any body cells, they're not irritating at all. These guys just basically grab on to antigens and then they get washed out. They don't really stick to anything. So number 3 tells B cells to make IgAs, which will clear antigens out of mucus. Basically, you find IgAs wherever you find mucus, like in your respiratory tract, your nose, your lungs. Also, there's a lot of mucus in your digestive tract too. So you find IgAs a lot, digestive system and respiratory system. And this is supposed to be what clears out things like cat dander and pollen and things. So what happens if your body, though, by accident, puts an E stem on these things? Let's say that these antigens or cat dander, which a lot of people are allergic to. But if the cat dander is sticking to IgAs, it's no problem. It just gets washed out of your nose. However, if the body by mistake starts putting the E stem on here, then these come apart. You don't have this situation. E's look like IgGs, right? IgEs just look like that. And this part can go stick to a mast cell. So then that switch was not so good. Then you get all these IgEs floating around with the cat dander, and that causes all kinds of allergic reactions. So the number 3 helper is pretty good. He tells the B cells to make more IgAs. So just so you are aware that there are some other options, and if you read a lot about the immune system, you'll see these mentioned, and you might not be sure where they came from. So there's where they came from. And hopefully you'll be able to use this as a general guideline that you can build on. You'll learn more information about some of these steps, but you'll know where that information fits into kind of the overall scheme.